the strongest radio allowable by law. Secrets will be revealed. Myths dispelled. From the studio gym where excuses never apply, it's Superhuman Radio with your host, Carl Lenore.
Welcome back to another episode of Superhuman Radio. I am donning my COVID-19 bandana that I can put over my face. It also comes in handy when robbing banks, but that's another lifetime. We have a great show today. You know, people love to say sugar is addictive. It's really not addictive in the clinical sense of the word. It doesn't do the type of things to the brain that addictive compounds do, but it clearly seems to drive consumption and habit forming. And we're going to be joined in a moment by Dr. Alexander Sisti to talk about why that actually may be. Before we do that, let's, of course, first pay homage to our title sponsor, a company that strives hard to keep you from eating too much sugar, and that's Legendary Foods. Legendary Foods has a bunch of great snacks, seasoned nuts, almond butters, and peanut butters that taste decadent, but all of them have very little sugar. Their tasty pastry, which is not depicted there, but I'll get a picture of it for the next show. It actually is a Pop-Tart with 9 grams of high leucine protein and less than 1 gram of sugar, and it tastes amazing. And if you go to the website eatlegendary.com and use the code SHR10, you'll save 10% off all of your purchases there. Show them some love. They are, in fact, a really, really great company. And then bring my guest on here and get rid of this. How you doing, Dr. Sisti?
I'm good. Thanks for having me on.
It's a pleasure. It's a pleasure. And especially because you're Italian. I like to hang out with Italian guys, you know? So there you go. Anyway, what I said at the beginning of the show has been going around the internet. There are people out there who say, you know, sugar is like cocaine. These are people who've never done cocaine, by the way. Sugar is like cocaine. It's addictive once you start eating it. And it's not addictive in the clinical sense because it doesn't change the structure or density of neurons in the brain in the area that it stimulates, but it clearly stimulates the brain. And it's probably as a result of evolution, right? Sugar was a good thing a couple million years ago because it was fast energy, it would keep you from starving to death and get you to move more and so on. So we probably looked for it. But it really isn't addictive. Can we clear that up first of all?
So the term addiction, I guess, it's like a psychological term that is rooted in human behavior. And to say that you're addicted to a substance, you need like this DSM criteria of it's causing lack of functioning, and there are all these sort of psychological behaviors that go along with addiction. But the sort of neurobiological root of it comes from the release of dopamine, right? Which everyone knows is this like the pleasure, the so-called pleasure neurotransmitter. But in that sense, sugar is actually a very powerful stimulant to release dopamine in the brain. And so if you just look at like the neurotransmitters that are released by things like heroin and cocaine and other addictive drugs, sugar is definitely able to cause the release of the same neurotransmitters. And maybe, in fact, those drugs are like artificial versions, like the original drug, the OG drug is sugar.
They're leveraging something that we have evolved to respond to. Exactly. Great point. Never thought about it that way.
Yeah. So, the term of addiction, again, is a little wishy-washy, but in terms of, sugar's ability to create pleasure and to create wanting and liking, and we call this sort of hedonic behavior, like pleasurable behavior. In that sense, sugar is extremely pleasurable, extremely rewarding. And I think that's why so many people have a hard time cutting it out of their diets.
Now, excess sugar intake has been linked to not only obesity, but obesity-related conditions like type 2 diabetes, which the population is plagued with today, but other issues as well that we're starting to learn. And it's associated with 500 million million, 500 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 million, 500
You know, one of the, I guess sugar has like a really interesting and rich history. And kind of, we can see what happened in the United States as the price of sugar goes down, the consumption of sugar goes up, and this sort of sugar-related health problems become more and more prevalent in our society. We can get into that in a second, but so many people, 70% of Americans are overweight, 40% of Americans are obese. Sugar is playing this sort of outsized role. But then as other countries in the world begin to, get more developed and, they start importing our Western lifestyle, our diet, they start getting McDonald's and, venti frappuccinos. And as the access to sugar becomes more widely accessible, then it just, starts affecting people all over the globe. And there's actually this, kind of like a historical trajectory, which actually first started in Britain, then the United States, and now is kind of spreading. I think Mexico right now might be the most obese country by proportion.
So let's talk about the relationship. We've heard a lot about the gut-brain connection.
Mm-hmm.
Now, your research, what led to your research? What preceded it that made you look at these questions?
Yeah, sure. So I guess at the outset, there's two main questions that we wanted to answer. The first question we've sort of been getting at, which is that throughout our human history, and our species just has this weakness, this insatiable desire to consume sugar. So about 200 years ago in the 1800s, when sugar was pretty rare, the average consumption was only about 10 pounds per person. But as sugar has increased in availability and decreased in price, as it's added to all sorts of foods, our ability to consume sugar has skyrocketed. So now the average American eats 124 pounds of sugar a year.
Wow.
Which is insane. Added sugars are about 15% of the American's daily caloric intake, just added sugars. And as I mentioned, a huge segment, almost the majority of our population is overweight, obese, diabetes, metabolic syndrome. And so there's this whole public health epidemic where sugar is playing like a crucial role. And so we wanted to understand, what is it about sugar? Unlike, for example, fat, like this example I sort of gave, you're at dinner, someone puts a big steak in front of you, you eat the steak, and the steak makes you feel full. It triggers this satiety reaction. But then at the end of the dinner, someone puts ice cream or something sweet in front of you, and then you just have this sort of unlimited capacity to consume sugar. So where does that come from? Why do we like sugar so much? And the second thing is, well, the most obvious answer to the first question is because sugar tastes sweet. And so, my lab, we study the sense of taste, and we have discovered many of the receptors and neural circuits involved in taste. And so the most obvious answer for why we like sugar so much is because it tastes sweet and we like sweet things. But
that nuance, we like sweet things, is a result of millions of years of selection pressure.
Yeah.
Because that was like if we look at substrates and fuel, that's gold.
Yeah.
Platinum. Maybe it's platinum, actually.
Actually, that's what sort of drew me to study the sense of taste in the first place. So with all your other senses, if I were to ask you, what auditory stimuli, what music do you like to listen to, or what do you find to be a beautiful painting? You and me and all your listeners, we're all going to disagree. And we can sit here debating for hours about what we like. But taste has this wonderful evolutionary hardwired logic where the things that taste good, so there's 5 tastes: sweet, sour, bitter, salty, and umami. And they break down into things that taste good and things that taste bad. And the things that taste good are sweet, which is typically like sugars. So sweet provides energy. Umami, which is the taste of amino acids. So that gives you protein, which you need to build your body. And low salt, because your body needs electrolytes. Whereas the things that taste bad, bitter and sour, tend to be poisonous, toxic, spoiled foods. So in actually the neurobiological substrate, all of these tastes, which my boss has worked on for 20 years, they travel in segregated sort of neural pathways and go to dedicated parts of your brain. So this information is always sort of separated. It's super hardwired, super innate, conserved across many species, and things that are sweet just immediately trigger this liking, wanting reaction, likely because, it gives you calories and energy.
Yeah, quick energy.
Right. But so that brings me to the second point, which is, so if sweetness was the only thing that could explain our attraction to sugar, then we have these things, these artificial sweeteners. Now, these it's sort of a mistake that they exist. They're chemicals that bind to the sweet taste receptor, and they're actually much more effective at stimulating your sense of sweet taste than any natural sugar. They're hundreds of times sweeter.
Right.
But if those chemicals could perfectly impersonate sugar, then they should have replaced sugar in our diets. So they were discovered over 150 years ago. And if those things like saccharin, aspartame, sucralose, if those could act as, or Splenda equal sweet and low, if those things could replace sugar, then they would have. But diet sodas have existed for decades, and they have not really decreased measurably the consumption of regular soda. We're still on that upsloping curve in terms of how much sugar we consume, even though we have something that's a much more potent sort of agonist for the taste system. So,
well, and I would offer from an evolutionary perspective that plants, the goal of the plant is to continue the species.
Mm-hmm.
And one of the ways that plants continue the species is to get other organisms or animals or bees to interact with them to help spread them.
Yeah.
And I would suggest that, and please, shoot me down here, that the intelligence in plants somehow figured out that these humans love sugar. If we could taste sweet, they'd pick us up and carry us places, and we would then proliferate to a greater degree. And that becoming sweet was actually an objective of plants.
Yeah, I mean, they devote a lot of their energy, the plants devote a lot of their energy and resources into producing, sugar-rich fruits, which kind of trick us into eating them, or sweet nectar, which gets bees to pollinate them. And so it's this extremely potent motivator that, it's not just in humans or in mice and rats. It's, flies, bees, worms,
deer, pigs, everything. Yeah. And I mean, if the purpose of delicious fruit, because keep in mind that fruit trees have been around way before commodes were invented and central sanitation. The purpose of the fruit was to get you to eat it, and then one day poop it out in this fertile, rich compound, mm-hmm. which is your excretion, where it could then grow.
Yeah. Yeah, it's like symbiosis, where the plant is producing sugar, which we will seek out, and naturally these ripe fruits only occur at a certain time of year, and they're super energy dense. So, going back to these hardwired neural circuits, we would expect that there are these hardwired pathways, so that that the few months of the year where you can get that ripe tomato, the ripe apple, you're gonna sort of seek it out, hone in on it, and try to consume it. But from the artificial sweeteners, we sort of intuited or guessed that maybe there's more to this sugar sensing than just your tongue and your sense of taste. So that's sort of what led us to look for other kind of neural pathways.
Because in fact, you point out in the paper that things like diet soda and artificial sweeteners that are used in coffee, while they may taste similar, and we know that glucose signaling occurs at the tongue. Mm-hmm. The body prepares for something. It goes, ooh, like the frontline guys say, hey, it's coming, and then the pancreas and everything else start to work. But we still know that even though they taste like sugar, the brain knows they're not sugar, right?
Yeah. So it's almost like the artificial sweeteners can trick your tongue, but they can't trick your brain. So we actually, we tested this directly. So, I think we have a lot of anecdotal experience. Anyone who's had a diet soda sort of knows it doesn't really, give you that same kind of feeling. and those sugar-free candies, they're really kind of garbage. But we wanted to test it in kind of like laboratory settings to really kind of hone in on what is it that we're preferring and how does it kind of work. So we actually set up these mouse cages where we do a sort of Diet Coke versus regular Coke challenge for a mouse, where we give it 2 water bottles, one with artificial sweetener and one with sugar. And initially they both taste sweet, right? So the mouse is like, oh wow, this one's really good. Oh wow, the other one tastes really good. But after about 24 hours, the mouse is only drinking the regular sugar. And then by 48 hours, it's like 90% drinking the sugar and barely touching the artificial sweetener, even though they both sort of still taste good.
Now, when we talk about artificial sweetener, there are a couple natural sweeteners, if you will, right? So sugar alcohol, obviously is one of them. Do we talk about sugar alcohol when we talk about artificial sweeteners?
Yeah, so I guess like my general definition of an artificial sweetener would be something that stimulates that sweet taste receptor without providing you like biologically available energy. So something that you can't metabolize. So something that you synthesize in the lab versus something that you could extract from a fruit, as long as it's, can't be broken down into ATP, then I would call that like an artificial sweetener.
Okay. So but basically you're saying any non-nutritive sweet thing is falling into this category. Yeah. So your research was done on rodents initially, correct?
Yes, because
describe how the study was designed.
Right. So we're trying to, like I said, we're trying to answer 2 questions. Why do we like sugar so much, and why do artificial sweeteners not really work? So we do this 2-bottle test, and we see that the mice love the sugar. This sort of confirms our anecdotal experiences. But the crazy thing is, in our lab, in studies of the taste system, we have made these genetically engineered mice that don't have a sweet taste receptor. So these mice are sort of sweet blind animals. They can't taste anything sweet. And when we do the same test with them, they also can identify and pick out the sugar. So what that means is there's something about the sugar that's working totally independently of this sweet taste pathway. So these animals can't taste anything, and yet they still like the sugar. So the sugar is acting somewhere else in some other mechanism. And so actually we thought that maybe it had to do with the sugar molecule itself. So not just the sweet taste receptor, but the sugar molecule. So we can give a sort of a fake sugar molecule. This doesn't really taste good, and it doesn't provide you energy, but the molecule, the structure looks almost exactly like glucose, and it has no energy. And then when we test that with artificial sweetener, we see that the mice also like the fake sugar. So what that gives us is mice can like sugar without tasting it, and mice can like sugar without even giving it calories. So there's something sort of special and unique about the sugar molecule that your body is somehow sensing. Actually we can infuse it into the gut too, and that also works. But you have this extra, we always talk about like sixth senses, but you sort of have this sixth sense that can help you identify and drive you towards the sort of sugar containing substances.
So in other words, the sweetness isn't the reward. The sweetener is the identifier.
Yeah.
But then the reward has to be there too. Otherwise we're like, oh, we got fooled. We're not going to keep eating that.
Exactly. Right. Like I don't know, sometimes in psychology, they talk about like the conditioned stimulus and the unconditioned stimulus, like the Pavlov dog, where the sweetness is actually like the bell that's being rung. It's the cue that tells you you're about to get sugar. Whereas there's another pathway that's sort of telling your body, hey, I actually got the thing that I needed. And so then, we did a whole bunch of experiments to try to figure out where's the sugar being sensed and how is it getting to your brain.
Okay, so to just kind of dive a little bit deeper, are these actual receptors in the gut that have like a mosaic that sugar docks in, or are we talking about microbes that produce chemical signals that get to the brain?
Yeah. So, there's a whole bunch of experiments in between the behavior and what you're asking, but we did want to know what is sugar triggering and how is sugar triggering the body to be able to recognize and prefer it. And what we knew, so how do you discover an unknown receptor? And what we knew is that both the real sugar and the fake sugar are transported into your body through the same sort of transport mechanism. So sugar has a number of physiologic roles. It can be metabolized into ATP. Maybe your body's sensing ATP. Sugar, when you eat sugar, it goes in your digestive tract, and then it actually has to be transported inside your body and gives you a rise in blood sugar. Maybe your body is sensing a rise in blood sugar. Or maybe there's some special receptor somewhere in your body that the sugar can trigger to drive these behaviors. And actually, this fake sugar molecule that we used is very elegant because it doesn't give you calories. It doesn't cause any hormones to be released. So no insulin is released. It doesn't affect your pancreas. But it is transported through this one transporter. It's called SGLT1. It is the main transporter that your body uses to move sugar from the outside of the body to the inside of the body. And so we hypothesized that maybe this transporter is actually playing a role as not just a transporter to move sugar, but as a sugar sensor in particular. And so, we did a bunch of experiments where we can give so this is actually really cool. So table sugar is a mixture of glucose and fructose.
Right.
And glucose is transported through SGLT1. But fructose, even though fructose has calories, it is not transported through SGLT1. It has a different mechanism. And fructose, if you do that artificial sweetener versus fructose behavior, the mice don't end up preferring the fructose. And we can actually look at the brain, at the neurons that are activated by glucose, and they're activated by glucose, they're activated by the fake non-caloric sugar, but fructose, which has calories, doesn't stimulate those same neurons.
But that would make sense because I think it's accepted that fructose is not readily and immediately available. It doesn't convert right to glucose. It has to be transported to the liver, technically stored, then reduced. So the body would go, that's great, we'll save that for later, but we need something for now.
Right. Glucose itself is the primary substrate. So, the whole goal of glucose is to generate ATP for your cells. And all the enzymes that do that need glucose specifically. So even though there are other sugars, fructose, mannose, ribose, everything has to be metabolically converted into glucose in order to provide your body with energy. So maybe it makes a little bit of evolutionary sense that this mechanism is specifically tuned to glucose in particular.
It's like you're searching your house for money, and you find a savings bond.
Yeah.
And you go, well, I can't cash that now, but I'll keep it because I'll be able to cash it later. Meanwhile, I'm still looking for legal tender that I can spend now. The body's doing the same thing. It's going, fructose is good. We'll save it, but that's not what we're looking for right now. So that makes perfect sense. So talk about the discovery of this.
Brain area.
Yes, exactly.
Right. So, this behavior that we're talking about, the fact that the mouse learns to identify the sugar over the artificial sweetener, it's not that simple, right? They're both of these things. For a mouse who lives in a standard lab mouse cage, he's only had water and mouse chow. Both of these things are the most delicious thing that the mouse has ever tasted before. Imagine living your whole life eating just like oatmeal and water, and then someone puts a Diet Coke in front of you. That's still the best thing you've ever tasted. So we figured that there had to be somewhere in the brain that's helping you to distinguish between these two stimuli. And they're both attractive, right? So if we looked at the brain areas that are important for taste, they would both light up for these things because they're both sweet. But what we were looking for was a part of the brain that is only activated by the sugar, that could specifically respond to the sugar and maybe drive this behavior. And so what we did is we, gave mice access to both of these solutions or either one of the solutions, and then we can look at their brains for this proxy for neural activity. And we found this area of the brain. It's called the NST, the nucleus of the solitary tract. And it's embedded deep in your brainstem. So people talk about like the reptile brain. And that's where this nucleus is. It's one of the most primitive, basic brain nuclei. But that kind of makes sense to us, right? Because we said that bees can do this, ants can do this, rats and mice. So it's this very primitive brain nucleus. It responds to sugar, but it doesn't respond to artificial sweetener. And it also responds to sugar in the knockout mice. So it doesn't depend on the sense of taste. And it responds to the fake sugar as well. So we figured that this brain area met all the criteria to be the sort of brain nucleus responsible for sugar preference.
Interesting.
Yeah.
So do you think that do you think that artificial sweeteners, or let's just say non-nutritive sweeteners, while they're giving people things that trigger the tongue sensation of sweet, that the body may actually go, I didn't get what I was looking for. I didn't get that sugar orgasm. I'm going to just keep eating until I get it. Do you think that non-nutritive sweeteners could actually drive overconsumption in searching for real sugar?
Yeah, that's a great question. We don't really have any data that specifically addresses that. I've heard that assertion kind of thrown around. I know that the artificial sweeteners don't trigger this sort of like these metabolic responses. But sort of from my perspective as a gut brain NST sort of expert, I know that the artificial sweeteners don't trigger the NST, which is really like, that's kind of telling your brain, I got what it wanted. So it's kind of
talk about the relationship between the brain cell and the vagus nerve. The vagus nerve is a very interesting wiring harness, if you will, that runs from the ends of the body into the brain.
Yeah, it's really fascinating. So next, after we found the part of the brain that is possibly responsible for this sugar craving behavior, we wanted to know where the information, how is it getting to the brain, because it's not from the tongue. And so this area is actually, it's very famous as being the primary recipient of information from the vagus nerve. And so what's the vagus nerve? Out of your brain, there's 12 pairs of nerves. They're called cranial nerves. They include your optic nerve for seeing, your olfactory nerve for smelling. Most of these nerves are contained to your head. They control, senses and movements and stuff like that in your head. But one of them, the vagus nerve, vagus comes from the Latin word that means to wander, like vagabond. And the vagus nerve, it comes out of your brain, deep in your brainstem, and it forms this neural pathway, this highway, where the vagus nerve goes to your gut, goes to your stomach, your intestines, and it also goes to your heart, your lungs, your kidneys, your liver. So really, it is the primary neural conduit through which your brain learns about your body. So, here we're talking about the gut-brain axis specifically, but it's part of this broader, what I would call it like the mind-body connection, which, sometimes a term is thrown around in like East.
Yeah, and it's kind of like this touchy-feely term, but this is the wiring harness of your body.
Yes.
When we think of a car, the wiring harness connects everything in the car to one another, and the vagus nerve kind of serves at that, and it's been implicated in a lot of functions and reactions in the gut and in the heart, as you point out.
Yeah, it's this really kind of underappreciated, unknown thing. I mean, I don't want to discredit the yogis that have been, practicing yoga and meditation for thousands of years. Like, they're sort of onto something, which is that there is some relationship between your brain and your body, right? Like your brain needs to know, what is my heart rate? What is my blood pressure? How can I control these things?
Is there food in my stomach? Because the vagus nerve controls digestive motility, moving food through the,
Exactly. And so now actually our paper and a few other recent papers are really kind of opening up this whole world of the vagus nerve. So, until about maybe 10 years ago, it was just sort of hinted at, but us and others are really trying to dissect what information specifically is carried through the vagus nerve. They thought it was only for vomiting and nausea. But what our study shows is actually that you can have very important positive information. So information about your nutrients, information about the distendedness of your stomach, information about the osmolarity. So that's the number of dissolved things like electrolytes and salts. So actually, it's this very complicated, wonderful neural pathway that's telling your brain and feeding right into this super primitive hardwired part of your brain that can very profoundly, we think, affect your behavior.
So the funny thing about that is 10 years ago, I did a show about a phenomenon called Romheld syndrome. Romheld syndrome is a reaction from the gut that causes tachycardia, palpitations, arrhythmia. And so people who suffer from Romheld syndrome will have a big meal, and then all of a sudden they'll develop palpitations, rhythm issues that they can feel. They go to the doctor, they get an EKG, they get an echo, and the doctor says, there's nothing wrong with you. They go home, they think they were crazy. 3 months later, they have this big meal, and it happens again. And what we discovered in that show, and since that show, is that some people, either through the phenomenon of the gut leaking into the vagus nerve, or because of structural changes in their body or structural components in their body, that when the gut blows up, it presses against the vagus nerve, that it's in fact the vagus nerve that's causing these changes in heart rhythm, and it's a mechanical reaction because the vagus nerve comes down along the esophagus and then literally attaches to the outside of the gut all the way around and then back down. So when you stretch the gut too far, the vagus nerve gets tweaked. And that's where Romheld syndrome actually comes from. And when people realize this, they can address it, don't eat too big of a meal, and they never have these episodes again. So the vagus nerve has always been very fascinating to me. And we also know that nurses, when people are in a clinical setting and they start to get anxious, the nurses can actually do something called a vagus massage, where they massage their neck and their heart rate will actually drop.
It's wonderful because it contains, in neuroscience, we talk about sensory versus motor information. So sensory information comes into your brain, motor information goes out and controls your body. And the vagus nerve is mixed. It contains both sensory and motor information. And just like you have the sort of patellar tendon reflex, just like, when the doctor taps your kneecap and your patella swings and you have these involuntary reflexes, the vagus nerve controls many autonomic or involuntary reflexes regarding your viscera. And so that you're talking about the carotid artery sort of bifurcates in your neck. And right in that bifurcation, you have a bundle of vagal nerve cells. And when you push on them, it sort of sends a signal that your vagus nerve all of a sudden thinks, oh my gosh, my blood pressure is through the roof. And then it does a reflex to your heart that says, yo, slow down the blood pressure.
Interesting.
My favorite illustration of this is there's actually a really funny YouTube video of these two Marines. And one guy karate chops the other in the neck. And he karate chops him right here in the carotid sinus of your vagus nerve. And the other guy just passes out immediately like a light. And so the vagus nerve has these very profound abilities to affect your behavior. The cardiac component was known, but the extent to which it could control our feeding behavior and give us pleasure through food, do these things like, eat 124 pounds of sugar a year and hijack into our dopamine reward circuits, that is kind of a total mystery. And, that's what we sort of unlocked here in our paper.
We're going to take a quick commercial break. When we come back, we're going to pick it up about what you did with these rodents and how you created these knockout rodents to test this theory, okay?
Sure.
Stay tuned. We'll be right back with more Superhuman Radio. And at the end of the show, you'll be able to leverage some of this information, perhaps, to control your own sugar cravings. I'm going to talk about mine when we come back also, because I deal with them as well. Do you remember those delicious toaster pastries you had when you were a kid? You know, the rectangular sugar-filled snacks? Well, guess what? Legendary Foods has just made low-carb toaster pastry. This is the first of its kind, and honestly, these things are amazing. They have 3 to 4 net carb, less than 1 gram of sugar, and 9 grams of protein. You can eat them right out of the wrapper or lightly toast them. The only question is, which flavor? Strawberry or brown sugar cinnamon? They're available at eatlegendary.com and Amazon.
New Mass Pro Synthogen X2 just upped its own legendary game. To distance itself even further from the rest of the pack, Synthogen X2 now has double the key active ingredients. If you've ever wondered what steroid-like recovery feels like, Synthogen X2 delivers. See why others compare it favorably to powerful bodybuilding drugs at synthogen.com. Mass Pro Synthogen. When you train with it, you'll gain with it.
I love beef, and if you love beef, listen up. I've discovered the best tasting beef in the world, and that's not an exaggeration, at Piedmontese.com. The Piedmontese breed is famous from Italy for being lean and unbelievably tender with half the fat and calories of traditional beef. Even typically tough cuts are tender when it comes from the Piedmontese cows. And for the first time ever, Piedmontese cows are being raised here in the USA. Get 2 free 10-ounce New York strips when you purchase $50 or more at Piedmontese.com with code SHR. Go to PIEDMONTESE.com and use code SHR today. You will never eat any other type of beef ever again. The precise way to gauge your recovery from workout to workout? Wonder if the money you're spending on your nootropic supplements are actually improving brain function. Maybe you're aging and you're noticing some changes in memory. Wouldn't being able to really test your brain be of great value? Well, now you can with great accuracy with the Brain. The Brain Gauge lets you test essential components of brain health and track your brain health history, and all in the comfort of your own home. Go to gaugeyourbrain.com and use code SHR for $150 off this amazing device. That's gaugeyourbrain.com and SHR for $150 off. Imagine if you had a digital twin, one that you could compare your own health and fitness outcomes to, one that showed you whether or not the things you're doing, food you're eating, or drinks you're drinking are actually working for you or against you. Well, now you can. The first ever advanced epigenetic saliva test that compares 20 million different data points of your DNA to help predict what is aging you faster or keeping you younger is being introduced to my audience at a 70% discount from the normal price. Go to seeds.md/epigenetic-test today to learn how to get your own digital twin that will help you take the steps to live longer and stay stronger. Don't wait, because this is a limited time offer not available anywhere. Once these tests are gone, they're gone. Again, go to seeds.md/epigenetic-test today to learn more.
This is the Superhuman Channel, where brawn and brains finally meet.
Welcome back. We're talking with Dr. Alexander Sisti about how sugar drives consumption. A lot of people have talked about this for a long time. His group has discovered really what makes us want more sugar. So talk about the research that you did with these little rodents here.
Yeah, so inside, like, to make a claim, like, we have discovered the neural circuit that is important for sugar preference behavior. We really use 2 criteria in science to prove causality: necessity and sufficiency. So the first is we have to show if this gut-brain pathway is really important, is really necessary for our sugar preference behavior, then the first prediction is that if we silence it or if we ablate the pathway, then it should eliminate the craving for sugar. So what we did, and this is using a suite of different genetic trickery, which is sort of unique to mice, which is why we use mice in the first place, is we can genetically target the sugar responding cells in their NST, in that brainstem area that is important for, that we think is important for liking sugar. And we actually inject a virus, a genetically modified virus, And the virus's purpose is to silence neural activity just in those cells. And when we do that, so now this mouse is normal in every way, except there is a small population of neurons in its NST that doesn't work anymore. They're not dead, they just can't communicate. And we take that mouse and we put that mouse into a cage and we give it the same sugar versus artificial sweetener test again. A normal mouse drinks, 10 times more sugar than artificial sweetener. These mice have no preference. They drink them exactly equally, which is totally insane. We have specifically silenced, we've silenced their attraction to sugar, but if we take these mice and we test their sense of taste, right, because we want to show that they still can taste sweet, we can give them artificial sweetener versus water or sugar versus water just as a sweetness test, and they're totally normal in that regard. So they drink the normal volumes, they can be attracted to sweet things, but specifically this behavior of liking sugar is totally ablated in these animals. And we can actually even do it in the vagus nerve itself. So I did a few experiments. Yeah, I did a few experiments where I can expose the vagus nerve in the neck of the mouse, both sides, because you have to silence both branches of the vagus nerve.
Right.
Whenever you're doing a neural silencing experiment, it's very important to do both sides because you can sort of compensate. And we can specifically silence the vagus nerve as well. And these animals also totally lose their attraction to sugar. So that's the first half. So silencing this gut-brain pathway or silencing these cells in the brain can totally ablate and make a mouse insensitive to sugar. The second half of our, criteria for causality is sufficiency, which is to say, is the activity in these brain cells sufficient to create a preference? And so these experiments were done by my collaborator, Wei. He's another graduate student in our lab, and he did this really, really beautiful experiment where he, using again sort of genetic trickery, we can target the sugar-responsive cells in the NST. But this time, he targets them, it's a receptor. It's like an activating receptor that responds to a drug. But this receptor doesn't exist anywhere in your body. So if you're a normal person and you take this drug, it has no effect. It's a completely inert drug. But you can take an excitatory receptor for the drug and you can introduce it specifically into the NST cells that respond to sugar. And so now it gives us a pathway to specifically activate these cells. And then he gave the mice access to 2 water bottles, one with a grape flavor and one with a cherry flavor. And one of these bottles is sweeter. Let's say that the cherry bottle is sweeter. And he gives them, an initial choice. And of course, the mouse likes the sweeter cherry flavor. But then he puts the drug specifically into the other bottle, into the grape bottle, and he sort of spikes it. But what the drug is doing is when the mouse drinks that bottle, the drug will be ingested into its system and it will go and turn on those cells in the NST. So we have made a sort of totally fake sugar, which the mouse can learn to associate with the cues, with
but it's still non-nutritive. But it just
not nutritive at all. This is it's like a drug that just
that does everything but give the nutrition value.
Yeah. So it is.
So what you really discovered here is the ideal non-nutritive sweetener.
In a way, we could use this, this neural pathway to hijack these, we found the neural circuit that you could hijack that's responsible for these sugar cravings. Now, the danger is that you could say, well, why don't I make something that I could give you? But that would just be like an addictive drug, right? Because
then you just crave that. Yeah,
exactly. You just.
But wait a minute. But from a food science standpoint, let's just connect some dots. So non-nutritive sweeteners aren't satisfying as sugar. So we continue to go back to sugar, but people consume way too much sugar. If you had a non-nutritive sweetener that leveraged this science, they could have one sugar-free candy bar and actually feel satisfied from it.
Yeah, so first of all, we explain why the artificial sweeteners are not good, and it's because of their failure to activate these cells.
Right.
So they have no effect. They affect your tongue, but they don't affect this gut-brain pathway. And so you're right that the huge sort of implication of this is that if we could figure out how to manipulate and change the activity of this gut-brain pathway, then maybe the ultimate goal here would be you could create an artificial sweetener that wouldn't just trick your tongue, but one that could trick your brain as well. And so that's like the sort of ultimate, gold.
Yeah, but see, this kind of research has been done before, right? Well, once we identify a receptor, we can make some sort of receptor modulator.
Yeah.
That docks in it and specifically activate certain portions of the mosaic of the receptor to give us the outcome we want. And if you can find, so theoretically, you could use one of the non-nutritive sweeteners now by adding a receptor modulator to it, where you get the tongue feel, you get the tongue taste, but the receptor is activated and your brain goes, oh, that must be sugar.
Yeah, so actually my boss, Charles Zucker, he discovered 20 years ago what the gene for the sweet taste receptor was. And so he used that information 20 years ago to try to develop the world's greatest artificial sweetener. But they failed because what they were trying to do is they made this, it was an amazing effort where what they could do is they could put the taste receptor into a cell culture system and sort of massively test millions of different compounds to try to find the ones that could specifically activate the sweet taste receptor. But the reason that those efforts were sort of doomed, and the efforts of Coca-Cola and Frito-Lay or whoever is trying to make an artificial sweetener that tastes sweet, is that you're missing a huge part of the picture. And so what our research does is it opens up this new avenue that, if we could instead think about targeting the gut-brain axis instead of just your tongue, that could be a road to to finding the holy grail of artificial sweeteners. Now, there's a sort of hiccup here. The problem is that we have discovered the pathway, and we have discovered the receptor that is used to trigger the pathway, SGLT1, that I mentioned earlier. The problem is that SGLT1 is the primary sugar transporter for your whole body. So if you were to, I actually, in the process of this,
it would have downstream metabolic effects if you block it. You're blocking like everything that sugar does for your cells as well.
Exactly. So we used CRISPR to generate a mouse that lacks this receptor. The problem is that receptor is also in every single cell that lines your gut that uses it to absorb sugar. And so now in these mice, sugar is actually toxic. They can't even eat sugar because it has so many bad consequences. So there's kind of a key missing piece.
That's where the selective receptor modulator would have to come in. The receptors have a mosaic of domains, so maybe there's a way to come up with a selective receptor modulator.
Well, actually, the fake sugar molecule that I mentioned earlier, MDG, it is not metabolized, but it is an agonist for this transporter. The problem is that because its structure so closely mimics the structure of glucose, that it actually in the long term, it's actually toxic to the mice as well, because it can be transported into your intestinal cells, but it can't be transported out. So it sort of clogs up and gums up the works.
Oh, that's not good. Yeah.
So if I were, the head of a major biotech company, but the way to sort of capitalize on this is really, we would want to find what the cells are, the specific cells that are responsible for signaling to the vagus nerve.
Or downstream, exactly.
Like in the middle of the stream. So we identified the receptor, we identified the vagal neurons, but we don't know which cells in the GI tract specifically express this receptor. And so
now what if the microbiome comes into play here? What if these chemicals, these molecules, are consumed by our microbes, and our microbes then poop out a neurotransmitter that then does the rest of the job? Then you could capitalize on that as well.
The microbiome is also super hot, on fire research field these days. The thing is that with our experiments where we're looking at single neuron activity, We can look at single neuron vagal activity, and I can flow glucose directly into the intestines of an anesthetized mouse and look at single neuron responses. And we know exactly what the signal is. And so we're sure that the signal is, glucose or anything that can stimulate this receptor. And actually, just as we did the necessary insufficiency with the NST, we could do necessary insufficiency with this receptor, where any ligand of it will stimulate these vagal cells. And actually, we did a pharmacological blocking experiment, and we blocked the receptor, and that totally eliminates the responses. But sort of what I was getting at is, what are the cells in your gut that communicate to the vagus nerve? So your GI tract, 99% of the cells that line your GI tract are enterocytes, which do absorption and digestion and metabolism. They're, line the brush border. They transport nutrients and do the work of digestion. But within those, there's 1% of the cells that are called enteroendocrine cells. And these cells have a more sort of neurological developmental origin.
And they're transmitting information.
Exactly. They secrete neurotransmitters. So like serotonin, norepinephrine, different signaling molecules, like serotonin, right? Like you hear SSRIs, which are a treatment for depression. There's actually as much serotonin in your gut as there is in your brain. And so there's this tiny population of cells. They're very sparse, but they have these direct communications to the vagus nerve. And perhaps by unlocking, which of those, because there are also different types that secrete different hormones, respond to different things, but understanding more about those cells and how they communicate, and those cells could certainly be the ones that respond to the bacterial metabolites from the, from your microbiome, right? Because there's the whole question, how are the microbes in your gut communicating to your brain? Well, we know what the pathway could very well be, right? The vagus nerve, which goes right into your, reptile.
Which is got to be where cravings come from, right? So so for years I didn't crave sugar, but loosened up my diet, and I start looking for sweet things after a meal, and it's truly a craving. It's like I'm not satisfied. Yeah, I'm not satiated until I eat something sweet after a meal. So it's clearly this phenomenon. So do we have any evidence that the terrain or the landscape of these receptor biogenesis increases in certain environments and decreases in certain environments? We know that going on a ketogenic diet completely changes the landscape of the gut. Is it possible to eliminate these receptors from the gut?
That is so with this specific receptor, this SGLT1 receptor, it's so vital for survival that to eliminate it entirely, like I said, would be have severe consequences. If you could modulate it or eliminate it in a specific population of cells, that would be much more targeted. But you raise a really, really excellent question of what happens to this circuit in disease states. So in an obese person, who has so many of these cravings, who's, has really developed a disordered pattern of eating where they, require so much stimulation to satiate their sort of neural cravings. Is this neural substrate modulated? Do they have more vagal neurons, less vagal neurons? Is their vagus nerve more sensitive?
Or does the affinity, to the receptor change where it constantly has to be fed again and again and again? Yeah.
But, in our study, we only really looked at, normal, but wild type,
non-disease state. Non-disease state.
So the one clear place that we could work to extend our studies would be looking at, animal models of obesity, how the substrates sort of adapt in that case. And there are also even animal models of eating disorders, like anorexia. You can make anorexic mice through different sort of mechanisms. And
you just have to nag them. You just have to nag them. Stop eating. Look, we have to take our last commercial break. When we come back, I want to talk about what your plan is, what the plan of your team is moving forward. Stay tuned. This is fascinating. I really believe that you're on the brink of coming up with the solution for an actually non-nutritive sweetener that will actually satisfy us. Stay tuned. Crank your muscle gains to new heights by transforming every gram of protein you eat into 3 grams with Masszymes. With 100,000 HUTs of protease per capsule, Masszymes increases your absorption of key amino acids, resulting in stronger, healthier digestion of proteins and certain vitamins that not only multiplies the impact of the protein you eat, but can also repair a damaged intestinal wall. Go to Masszymes/SHR for 10% off. That's MASSZYMES.com/SHR. Whether your goal is to build muscle or burn fat, you'll find everything you need at Redcon One. Need help getting a good night's sleep? Try Fade Out, or the most popular pre-workout supplement on the market today, Total War. Sign up for their new Transformation Challenge and win $10,000, or shop for apparel that people at the gym will know that you are serious about your training. Need a testosterone booster that works? Check out Boomstick. Whatever you need, you'll find the best quality supplements on the market at Redcon One. Go to redcon1.com. That's REDCON, the number 1.com, or go to superhumanradio.net and click the Redcon One banner ads today. How often do you sit with your laptop right on your lap? How much time do you spend on your cell phone? Are you in a technology-packed office Monday through Friday? Are you worried about this type of radiation? Now there's something you can do about it. GetLambs.com. This radiation has been linked to infertility in men, glandular tumors, gut microbiome dysbioses, and impaired sleep quality. Now you can provide 360-degree protection to at-risk parts of your body with radiation-proof apparel from GetLambs.com. Comfortable, breathable, and 99% effective. Go to GetLambs.com and use coupon code SHR for 20% off your order of $100 or more. That's getlambs.com, GETLAMBS.com, and code SHR. Hey, this is Carl. For 14 years, you've heard me talk about Can-C eye drops, and they being the reason that I do not need reading glasses at now 61 years old. But I regularly get emails and messages from people who've been using Can-C and having some amazing results. Recently, I got an email from a fellow named Chad, who, because he was on dexamethasone eye drops for over 6 months, developed a cataract. Can-C eye drops actually reduced my cataract to the point where even my doctor has a hard time finding it. I will never stop using Can-C eye drops twice a day. I've been using them since 2008, he says. And you should be too. There is no better way to keep your eyes healthy and seeing clearly than Can-C eye drops. Go to wisechoicemedicine.com today and get on board, and we will both be looking into the future with very clear vision.
Men and women, you've heard about hormone optimization. Do you feel like it's something you want to look into? RenewLifeRx.com is the place to start. Their doctors can help you with the solutions. RenewLifeRx.com has a simple process for lab work, consultation, and taking a deep dive into where your hormone levels can be improved. Superhuman Radio listeners get 30% off your initial lab work and consultation. Go to RenewLifeRx.com to schedule your no-obligation phone consultation today. Feel younger, get in better shape, and be more productive at RenewLifeRx.com.
You're listening to the Superhuman Channel. We're ripped and we're ready.
Welcome back. We're talking with Dr. Alexander Sisti. We're talking about maybe a pathway to finally the most effective non-nutritive sweetener ever. And this could be a game changer for so many people. You know, we're all driven by taste, but it turns out we're driven by much more, much deeper evolutionary edicts and gifts that make sure we don't get fooled. Because let's face it, our drive to wheat occurred millions of years ago because we knew that this was a high form, high delivery rate of energy. And our body probably got fooled numerous times, and it affected us. And we evolved to have secondary mechanisms put in place as check mechanisms to make sure that what we tasted, was really what we thought it was. So going forward, your team is planning on doing some amazing things, right?
Yeah, I mean, I would say that there's kind of two avenues going forward. The first, as you mentioned already, is looking at how these neural circuits and substrates change in different disease states like obesity, but there's also mood disorders, and there's lots of other psychiatric disorders as well that can give you sort of changes in your appetite and eating behaviors. And this pathway, maybe it's not the one that's affected in depression, but it could result, it could be a new way to kind of intervene on the brain and give you a sort of new pathway to, if we could modulate these primitive parts of the brain through the gut, it could give us a new sort of therapeutic target for other kind of diseases. And the second thing is also moving beyond sugar. So we've shown how important this gut-brain circuit is for the taste of sugar, but there are other macronutrients that our body needs, proteins and fats. And so do other nutrients also sort of activate this pathway? And then finally, as you mentioned, is there a way, by the way you would do it is by identifying the cells that express this SGLT1 receptor and which communicate to the brain through the vagus nerve. Is there a way that we could find some other receptor on those cells or some other way of activating or silencing them to more directly sort of reduce our cravings for sugar, eliminate that? Ideally, we're not there yet, but eliminate that 15% of our caloric intake. I'm not saying that we're at the stage of curing obesity, but this gives us a totally new way of looking at this problem of sugar overconsumption and a very specific anatomical substrate that we could try to intervene on and study further to address these huge issues facing our society. So
we know that fat seems to stimulate satiety. We know that protein seems to stimulate satiety. I, as a former 330-pound obese person, I don't remember sugar ever. I don't remember ever eating sugar-based things and going, I've had enough. I usually ran out. There was none left, so I just stopped. Is there a connection between the lack of satiety and this unique molecule of sugar?
The way that I think of satiety, just from my sort of basic understanding of physiology, is that proteins and fats can actually trigger the release of this hormone CCK, cholecystokinin, from your small intestine. So there are enteroendocrine cells in your small intestine that respond only to proteins and fats and cause the release of this hormone CCK. And CCK can act on your brain to reduce the feeling of satiety.
Hunger.
Reduce the feeling of hunger and produce satiety. And actually sugar does not, this is not our studies, but it's just known not to cause the release of CCK. So sort of from my perspective, the sort of lack of sating effects of sugar come from that, in part the lack of the hormone release of CCK.
And the lack of the hormone release has to come from an evolutionary process where we didn't get that much sugar. If we came upon a bee's hive and we would feast on honey that day, but the rest of the time we were chewing stuff. I mean, there was not a lot of sweet. Even, I had this, and I mentioned this so many times, and I think the people who have been listening to my show for the past 14 years get tired of some of these things, but Dr. Leslie Aiello, who used to run the Wenner-Gren Foundation, we had this talk when the paleo diet became popular. She says, we'll never be able to eat like our paleo ancestors because apples tasted like turnips. They weren't these sweet variations we have today. You could never get enough sugar from eating apples like you can get from eating one today. Yeah. So it's probably a result of the fact that sugar just wasn't that abundant.
Yeah, I guess, we have these underlying biological, I don't want to say vulnerabilities, but we evolved in certain.
Well, in modern times, they're vulnerabilities.
Yeah, so we have all these neural circuits to seek out and drive the consumption of calories in what was usually a very energy scarce landscape. And now that we find ourselves in an energy poor landscape where like I could get a McDonald's Big Mac for $3 or $5 or whatever. Now those things that we have had evolved to be protective are now coming back kind of to haunt us because we don't have any breaks really on sort of consumatory behaviors.
Yeah, and in fact, when the discovery of the thrifty gene was actually an asset a million years ago.
Yeah.
If we were able to survive on less and thrive, we were better suited for the environment that we evolved in. I mean, the time that we are in modernity, modern agriculture and all that sort of stuff, this is a blip on the timeline of our existence. So this is fascinating stuff. Will you come back as you learn more about this?
Sure, I'd be happy to.
Yeah, this is really great. And I hope that you guys discover a way to harness your science and make non-nutritive sweeteners not only as satisfying to the tongue as they are today, but satisfying to the brain as well. So people feel like they can eat one and be satisfied. That would be a wonderful thing, I think. A gift. It would be a gift to humanity. It would.
Yeah, I think so.
Listen, thanks for being here today.
You're welcome. Thank you very much for having me.
Take care. We're going to take one commercial break. And when I come back, I'm going to tell you about some phenomenon that I'm experiencing that I can blame on the coronavirus lockdown. I want to see if some of you are experiencing the same thing. So stay tuned. We'll be right back with more Superhuman Radio. Don't go away. You've heard about blood flow restriction training since 2006 on SHR, but you're still on the fence. Well, here's the push. BFR is more effective at building muscle than anabolic steroids. That's right, I went there, but it's because it's the truth. My experience with the Be Strong blood flow restriction system is proof to me, and now I'm asking myself why I waited so long. You'll see undeniable changes in the targeted muscles in days and weeks like nothing you've ever tried before. I will never stop using them. Give Be Strong a try. Go to bestrong.training/super-human and use code SHR for 10% off.
Quest Nutrition makes bars, cookies, chips, and pizzas out of complete dairy-based proteins. Our products minimize net carbs and sugar without sacrificing taste. Each delicious chocolate-flavored chip, cookie chunk, and crunchy crumble is custom-made to maintain Quest macros. It's time to enjoy foods that work for you, not against you. It's time to enjoy your Quest.
7,451. That's how many people kick the bucket, buy the farm, or cash in their chips every day in the United States. Yep, that means 5 people are going to meet their maker during this commercial. And no, our commercials are not the cause. Half of those who punch the clock for the final time will do so without life insurance. Call Big Lou at Term Provider before you are number 7452. If you're a 50-year-old male, a tad porky with a touch of diabetes, $1 million of term life insurance may only cost about $200 a month. With more verified 5-star reviews than any other Lou on the radio, Big Lou has saved thousands of people thousands on their term insurance premiums. Stop procrastinating and call Big Lou today at 800-560-0301. 800-560-0301. Remember, Big Lou's like you. He doesn't want to be number 7,452. Call 800-560-0301 or biglou.com.
Ever wish there was a precise way to gauge your recovery from workout to workout? Or wonder if the money you're spending on your nootropic supplements are actually improving brain function? Maybe you're aging and you're noticing some changes in memory. Wouldn't being able to really test your brain be of great value? Well, now you can with great accuracy with the Brain Gauge. The Brain Gauge lets you test essential components of brain health and track your brain health history, and all in the comfort of your own home. Go to gaugeyourbrain.com and use code SHR for $150 off this amazing device. That's gaugeyourbrain.com and SHR for $150 off. Do you remember those delicious toaster pastries you had when you were a kid? You know, the rectangular sugar-filled snacks? Well, guess what? Legendary Foods has just made low-carb toaster pastry. This is the first of its kind, and honestly, these things are amazing. They have 3 to 4 net carb, less than 1 gram of sugar, and 9 grams of protein. You can eat them right out of the wrapper or lightly toast them. The only question is, which flavor? Strawberry or brown sugar cinnamon? They're available at eatlegendary.com and Amazon.
Spit that out right now. This is the Superhuman Channel.
Yes, that's right. We are now available on your Alexa smart speaker. Just walk up to it and say, Alexa, play Superhuman Radio Podcast. And you can listen to the show in the comfort of your own home. And soon the live channel will be back up and updated with all the old shows and run around the clock. And we'll have a separate Alexa channel for that. So get ready. We're still working some of the bugs out of the Alexa channel. You may have to say, Alexa, play Superhuman Radio Podcast. But we're going to bridge the word superhuman for some people who say it faster. So it should open up for you and you can play that. I want to answer a question first for Ivo Sue. I didn't want to put this up during the show, Ivo, just because this is probably outside of the good doctor's wheelhouse. But I'll tell you why this is. This is an interesting phenomenon. CO2 is given off by plants, and O2 is taken in by plants. No, I'm sorry. CO2 is taken in by plants, and thank you. I got it backwards. They're the opposite of us. And carbs and sugar are what attract animals to eat the plants and then take them to various places and poop them out or drop the seeds or whatever, because we actually are like we're like the honeybees of many plants, because we take them, we eat them, and we help them grow in different places. And so the more CO2 in the environment, that means that technically there are less plants, and that's a signal to the plant that they need to upregulate their production. So this is one of the reasons. The sugar is a way to attract animals like us to the plant, to get us to carry them. But you're right. The more CO2 there is, and that's why they use CO2 even in growing marijuana, because CO2 increases any of the quote unquote reward molecules of the plant. In the case of marijuana, it's THC. The more that's how they've gotten marijuana plants to be 32% THC now. It's ridiculous. It's like 10 times more than when I was a kid, because they grow it in enclosed rooms and they enrich the surrounding air with CO2. But yeah, and that's because those are the things the plant upregulates in order to get animals to come and play with it and carry it around, let's say. So I haven't been to the gym now. I don't know. I guess maybe for around 49 days. I've been training at home, but not a lot. I've only trained a few times a week at home. I just can't get into it at home. I'm sorry. I'm one of those guys that I prefer going to the gym and training. I prefer seeing my friends and talking, but also I don't have the equipment at home to truly load my muscles the way I like to load them. You know, it's that heavy weight that keeps me feeling good. And since I've been away from the gym, I don't know if you have this experience too, but I feel horrible. I have pains that I've never had before. This is the longest I've taken off. Even when I had surgeries on my foot, I still trained upper body. I still trained the other leg. You know, I still was active, probably more active than the average person in the gym. But this is killing me. I'm starting to understand why so many people are dying from being locked down. So in Louisville, Kentucky, murders are like they've quadrupled. And the local administration is saying, oh, it's because people are undirected. They've got time on their hands. That's not what it is. If people are going stir crazy, number one. Number two, they're feeling oppressed. Whenever you oppress people, they strike out. You tell people they can't do something, they're going to find a way to do it. But also, a $1,200 stimulus check is nothing to somebody who barely gets by on $3,000 a month, a single mom with 4 kids. And so I think we're going to start seeing more and more violent crimes, more and more lootings and robberies and home invasions and so on. But also, I don't feel good mentally. I'm not training, and I just don't feel good. I actually, for the first time in probably 20 years, feel old. I really do. And it's just because I haven't been in the gym getting my medicine. And my medicine happens to be very intense training for me. I'm not going to be as strong as somebody else out there. I'm definitely not as strong as I was when I was in my 50s. I was really my strongest in my 50s, by the way. But I really am feeling this, and I'm starting to think to myself, we're doing a lot more harm than good with this lockdown. A lot of people are complaining about not feeling good. There's a lot of people saying that people with mental illness and depression are suffering. Hell, some of us who didn't have mental illness and depression may come out of this with mental illness and depression. If you're feeling the same way, I'd love to hear from you. On air at superhumanradio.net is the way to communicate with me. I'm hoping that we can start. And the worst part is most of these governors They're misguided. If you've listened to any of my shows the past 2 weeks, at the end of every show, I do a little thing about COVID and how guided this whole approach is. And that's my opinion. You could have a different opinion. We could both coexist in the same room. That's not a problem. But this is misguided what they're doing to us right now. And the only people that should be in lockdown are the people most susceptible to dying from this, which if you look at the age stratification now, we have really good data now. We have tens of thousands of deaths. The people that are dying are in their 80s, and the large majority of them are in nursing homes. The large majority of them are in senior facilities. The large majority of them are actually at the end of their lives. So those are the people that need to be protected. People with strong constitutions and good immune systems, not so much. But I am just, if I don't get into the gym today, that was a Freudian slip. If I don't get into the gym soon, I don't know how I'm going to feel. And our governor here in Kentucky thinks that gyms are, they're not important. They're just not important. I don't know. But if you are feeling this, I would love to hear from you at onair@superhumanradio.net. And also, if you want to be an investigative reporter, I've got a couple projects I'd love to put some people on, and you get on the air with me and talk about your findings. Fascinating show from Dr. Sisti. I would love to learn more about a sugar substitute that actually satisfies all of the effects that sugar has on the human condition. That would be a big game changer for any company that's trying to make food products that are healthier, really. All right, that's it for today. Let's see, tomorrow's Thursday. I think we have a Renew Life RX show tomorrow. And then Friday, Dr. Elizabeth Yerth is coming on to talk about a couple peptides, GHK, CU, and another GHK, and what they're good for. I know they regrow hair, so anybody who's losing their hair is going to want to listen to Friday's show. It's at a special time. It's at 2 p.m. instead of 1. So check us out. See us then. Please share the show. Please share the show. I keep asking people to share the show. Just email it to friends. If you think the show is good, if you think the information is important, please send it to people you know. All right, see you tomorrow. Thanks for listening today and watching.